Fertilization occurs when sperm penetrates the egg. Only one sperm will be successful at penetration. Once it has penetrated, the egg undergoes changes to prevent the entry of another sperm. Sperm deposited into the vagina go through a final maturation step called capacitation in response to some chemicals and enzymes that are present in the vaginal secretions. Without this step, it's actually not capable of penetrating into the cell. To get into the cell, it actually has to chew through, and that's where the acrosome comes in handy. It's got to chew through the outermost cells and through the zona pellucida. Once it has entered into this space, okay, I want you to notice from this picture first that we don't have a nucleus. So even though we saw nuclei drawn on previous pictures, those pictures were inaccurate. The nuclear envelope is not present within the ovum, and as you note, the ovum is suspended in that second meiosis 2, second meiotic division, specifically at metaphase. Metaphase 2. When the sperm enters, we essentially have two sets of chromosomes. The chromosomes that are present within the sperm, and I'm just going to kind of doodle those in, and that will be housed in the head of the sperm, which is what actually makes it into the cell, and then the chromosomes shown here as well. Here we can actually look at each step of fertilization in more detail, and I want again to illustrate some things that I want you to look at and study and be aware of. First, I want to point out that first polar body. Remember in meiosis 1, one of these, the first polar body is created. One cell hogs all of the cytoplasm and organelle, and the other cell is essentially just a nuclei. The nucleus of the sperm is injected, I guess would probably be a good way to describe it, into the ovum. And we actually at this point have a nuclei right there. And now we're going to have a continuation of meiosis 2. And so in this case, we're actually in anaphase 2. And that completes the process. And one of these, and it's kind of random, it doesn't matter necessarily which set, but let's go ahead and say my green set, is going to be expelled and becomes my second polar body. And so these polar bodies tend to hang on to the side of the ovum. And at this point, notice we have a second nuclei, so we actually have two nuclei here. And then those nuclei are going to fuse, which gives us our zygote. Okay? They fuse to form the zygote nucleus. The reaction that prevents another sperm from penetrating is called the cortical reaction. Okay, and that's just a vocabulary term to be aware of and to know. And it simply just changes this outer layer, this zona pellucida, so that we cannot get fertilization with a second sperm. Okay. Once this zygote or the zygote nucleus fuses, we're back to being a diploid cell, 2N. And from this point on, embryonic development is going to proceed by mitosis. Fertilization must occur within 12 to 24 hours of ovulation. So if you remember your LH surge, okay, LH surge, approximately 12 to 24 hours after that surge, we get ovulation. And that oocyte enters into the fallopian tubes. Around that 24 hours, within that once ovulation occurs, that oocyte will only live for 12 to 24 hours before it breaks down and decays. If a sperm fertilizes it, though, then it will continue to live, and it will continue to, at that point, it'll begin to develop, and it will very rapidly divide and produce the embryo. Fertilization usually takes place in the distal part of the fallopian tubes. Now, just statistics-wise, because this is a little bit mind-blowing, if we assume that 2 million sperm are ejaculated, only about 100 of it make it through to the egg. The environment, the vaginal cavity, that environment is actually very acidic, and even though sperm, or more precisely semen, has buffers in it to buffer that acidic environment, the reality is that can only take it so far. And these sperm tend to die out. Only a small fraction of it will actually make it into the distal part of the fallopian tube to fertilize the egg.